Hello, I'm James Harding, Assistant Attending on the Gastrointestinal Medical Oncology and Early Drug Development Services at Memorial Sloan Kettering Cancer Center. Thank you to the Lancet Oncology for publishing our work on Zanidatamab for HER2-amplified advanced biliary tract cancer patients, and for the opportunity to present these data today. Biliary tract cancers, which include intrahepatic cholangiocarcinoma, extrahepatic cholangiocarcinoma, and gallbladder cancer, are uncommon, accounting for about 1% of all solid tumors. Most patients present with incurable, locally advanced, or metastatic disease. For such patients, the standard of care in the first-line setting is gemcitabine and cisplatin, plus the anti-PD-L1 antibody durvalumab. After failure of first-line therapy, the current treatment paradigm is either additional cytotoxic chemotherapy or treatment with precision medicine-based treatments based on genotyping of the patient's tumor. It is now clear that HER2 overexpression is observed in a subset of biliary tract cancers. Estimates range from as high as 20 to 30% in gallbladder cancer and extrahepatic cholangiocarcinoma to as low as 4 to 5% in intrahepatic cholangiocarcinoma. Single-arm prospective studies have indicated that blocking HER2 signaling in patients with HER2-positive BTC with monoclonal antibodies, tyrosine kinase inhibitors, antibody-drug conjugates, and combination treatment has anti-tumor activity. However, no therapies have received regular approval in this disease. Zanidatamab is a bispecific antibody that binds to two separate HER2 epitopes, leading to inhibition of HER2 signaling and anti-cancer activity in preclinical models of cancer. Zanidatamab has shown a manageable safety profile and encouraging anti-tumor activity in patients with HER2-expressing biliary tract cancer in a phase 1 study. To further evaluate Zanidatamab in patients with advanced or metastatic HER2-amplified BTC, the global multicentre phase 2b Horizon BTC-01 study was conducted with the primary objective of defining the confirmed objective response rate by blinded independent central review. Secondary objectives included duration of response, disease control rate, progression-free survival, overall survival, and safety. Planned subset analysis explored the impact of HER2 positivity by IHC and other clinical variables on objective response rate. Patients with HER2-amplified BTC confirmed by central testing with in situ hybridization enrolled into one of two cohorts. Cohort 1 enrolled patients with 2 or 3 plus IHC by HER2, termed the HER2 positive cohort. Cohort 2 enrolled patients with HER0 or plus 1 by IHC. All patients received Zanidatamab 20 milligrams per kilogram intravenously every 2 weeks. The study completed enrollment in March 2022 with 80 patients in Cohort 1 and 7 in Cohort 2. At the time of the data cutoff on October 10, 2022, 18 patients remained on treatment. In Cohort 1, 65% of patients were Asian, 51% had gallbladder cancer, 78% had HER2 IHC3+, 89% had AJCC stage 4 disease, and all patients had at least one previous line of therapy ranging from 1 to 7. In Cohort 1, the HER2 positive cohort, we observed an objective response rate of 41.3%. One patient achieved a complete response, 32 a partial response, 22 with stable disease, and 24 with progressive disease. One patient was not evaluable. Most patients had some degree of tumor shrinkage, and responding patients often had a rapid response with a median time to response of 1.8 months. Responses were also durable, with a median duration of response of 12.9 months at the time of the data cutoff. Importantly, pre-planned subset analyses showed that the objective response rate was similar for nearly all subsets. A striking finding is that patients with HER2 IHC2 plus tumors had a lower objective response rate of about 5% versus 50% for those with IHC3 plus tumors. Such observations have been observed in HER2 positive gastric cancer, and further study in this population is required and planned. In cohort 2, patients with IHC 0 or 1 plus, no responses were observed in 7 patients. Other secondary time to event endpoints, such as progression-free survival, which was 5.5 months, and overall survival, which was immature, require further follow-up. The toxicity was similar to previous reports with Zanidatamab, and the typical on-target HER2 adverse events, including diarrhea, nausea, and vomiting, as well as asymptomatic ejection fraction decrease, were observed. Infusion-related reactions were observed at low grade, and all patients received mandatory pre-medications. No grade 4 or 5 events were observed. In summary, our study demonstrated that Zanidatamab led to a confirmed objective response rate of 41% in patients with HER2-positive BTC who progressed on prior therapy. The agent was safe and tolerable in this treatment-refractory population. These data add to the growing body of evidence that targeting HER2 in BTC has anti-cancer activity and is probably clinically meaningful. Correlative studies will nominate markers of response and resistance to Zanidatamab, while ongoing and planned studies will seek to confirm the observed activity on the single phase 2 study, and will further define the efficacy in this unique patient population. Thank you for your interest and attention. Again, thank you to the team at the Lancet Oncology. Thank you to all the co-investigators, study sponsors, and of course the patients and families who participated in this important study.